Hello, everybody. I am the president of the French Society for Cell and Gene Therapy, and I'm happy to present to you a new webinar on AAV vectors, applications, and production. And this particular webinar is not fitting anymore in the series of the gene editing webinars, and is also with the support of Pfizer Maladies Rares. So today I'm going to present you the next speakers, which is Gijsbers from Leuven and Oumeya Adjali. So Rick, he's a trained bioscience engineer and is an associated professor in the Department of Pharmaceutical and Pharmacological Sciences at the KU Leuven. He's also heading the Laboratory of Viral Vector Technology and Gene Therapy, and his lab focuses mainly on viral vector manufacturing and technology development and applications. These developments include also, for example, cell models and gene therapeutic approaches for rare genetic disorders, like, for example, STAT1 gain of function. In addition, he's also responsible for the management and the scientific innovation of the Leuven Viral Vector Core. It's a platform for target identification and validation to develop both cell and small animal models using different viral vector-based technology platforms. So please, Rick, you have the floor and you can start.
Thank you very much, Alice, and welcome all to this webinar on gene therapy and viral vectors, where I, together with Professor Adjali, will try to fill you in on how we can re-engineer viruses into viral vectors, and then to treat genetic disorders, how we will scale them, and how we do quality control. So I will try to fill you in on gene therapy and viral vectors. What are viral vectors, and what are they actually, and what are ATMPs? To start with, ATMP stands for Advanced Therapy Medicinal Products. And these are medicine that are defined for human use based on genes, cells, or tissues. And they're providing groundbreaking new opportunities for the treatment of disease and injury. And this is how the EMA, or the European Medicine Agency, actually defines these. So here you see a different cartoon depicting what ATMPs are. And you can discern the somatic cell therapies, tissue engineered therapies, gene therapies, and then the combined therapies. And today we will mainly focus on one subset, namely the gene therapeutic medicines. I will, in the first about 20 minutes, discuss briefly the toolbox. It's like, what is a viral vector and how do we generate a viral vector starting from a parental virus? But to me, it's also very important that you realize that this is also a story of people. So what you see here are different people that suffer from rare diseases, rare disorders, for which gene therapeutic medicines have been developed. Some of them are currently already on the market, but just to show you that this is a field that is rapidly moving and for which we need additional hands to help solve the problems that we encounter. I also indicate Rare Disease Day, which next year will be again on its proper day, namely the 29th of February, where we actually have a special day for these patients. Now, the goal of my talk is also to provide you some insights, and but also to show that ATMPs, cell and gene therapies, are no longer science fiction, and that these slowly, because it's taking time, or it has been taking time, but they really are now a clinical reality. Just to give you an example, what you see here are the gene and cell-based products that are worldwide on the market. And this was Q3 of 2021, with about 20 gene therapies, 15 RNA therapies, and more than 50 cell therapies available worldwide. And this year in Q3, these have been raised to 27 gene therapies, 26 RNA therapies, and more than 60 cell therapies that are on the market. What is more is what is in the pipeline. With more than 2,000 gene therapies in the pipeline, with even, imagine only 10% of these will reach the market in the next 5 to 10 years, that will be a tremendous amount of new therapies that will be available to treat patients in need. Now, how can we make gene therapy work? In order for gene therapy to be functional, we actually need 3 critical elements to have a gene therapy or to design a gene therapy for a particular disorder. Firstly, it is, and this might look trivial, but it's a very difficult one, and it's something that only came about in the last 30 to 40 years. It's the development of molecular medicine, understanding the underlying molecular mechanism of a disorder, the fact that it's a genetic underlying problem, for example, is key in order to develop a gene therapeutic approach. Second, we need to know and have a good in-depth knowledge on the specific cell or tissue that we need to target, and we have to understand the target cell and tissue biology. And thirdly, we have to, once we know these two, we have to choose a way how to provide the specific therapy. Providing gene material to the brain, for example, will be different than providing it to the blood. So in the end, only when all these 3 specific elements have been elucidated, we can develop a gene therapeutic approach. And this boils down in 3 different steps. First of all, we have to define the package, the delivery of a genetic information to cure, halt, or prevent a disorder. Second, we will have to define a specific delivery route. Thirdly, we have also to define in which way, which kind of vector or which kind of vehicle we're going to use to transfer this genetic information. Let's take this step by step. So first of all, we have to understand the problem. In healthy people, we have chromosomes. On these chromosomes, we have the genetic information with genes, and these genes get transcribed and translated into functional proteins. A functional protein is a happy cell with a normal function, and these people and cells and tissues are healthy. However, we also know that people suffering from a genetic disorder, they mostly have errors or a lack of certain base pairs or mutations in certain base pairs. This results in the lack of a functional gene, which may result in either not sufficient protein, no protein at all, or a protein that is altered in its fold or in its function. This results in a suboptimal or an absent function, which will lead to disease. Secondly, so for these kind of diseases, in theory at least, we can provide a correct version of the genetic material. The question then is, how can we deliver this package to the cells? So, instead of using drugs and surgery to cure these cells, and thus the patients carrying these cells, we will now use genetic information to prevent or halt the specific disease. And there are different approaches that we can explore. We can replace the disease-causing copy of a gene, or we can add a functional copy of the gene. We can silence specific versions of a gene when that would be needed, or we can even edit it. And depending on the kind of disorder, we will have to choose either of these applications. Secondly, we have to choose whether we're going to treat the patient directly in vivo, and depending on the target tissue and cells, that will be defined, or the specific target cells and tissue will define that. As you can imagine, a brain is not that easily taken out of the person and then placed back in. And based on the target tissue, we will either have an in vivo direct delivery method or an ex vivo cell-based delivery method or tissue-based delivery method, where the tissue or the cells of interest can be taken out of the patient, treated and manipulated ex vivo, and then placed back into the patients. Thirdly, we have to define how are we going to transfer this genetic information. So which vector are we going to use? Two types of vectors can be discerned. On the one hand, non-viral vectors, such as plasmids or plasmids with polymers or liposome combinations. And the second option are virus-based vectors. Like in this webinar, we're going to continue with the virus-based vectors that are presented here as their parental, the parental viruses. Now, based on the parental viruses, different viral vectors have been developed that all come with their specific features. And it will be key when you try to develop a gene therapeutic approach that you try to use the best vector available to carry your specific payload and to transfer it to your specific target cells of interest. Now, it's very important to realize that at the moment, even though we have quite a nice armamentarium to transfer genetic information, there is no such thing as an ideal or a perfect vector. We will always, as scientists, but also as MDs treating our patients, have to make a trade-off between the different features of our viral vectors to transfer the correct information into the cells and tissues of the patients we want to treat. As an example, I will walk you through how we convert a virus into a viral vector. In this case, how we develop a recombinant adeno-associated viral vector starting from the wild-type AAV virus. So first of all, it's very important, as depicted here in the cartoon, an AAV virus is carrying a protein coat on the outside and a single-stranded DNA on the inside. And before we can convert a virus into a vector, we have to fully understand the genetic makeup of these viruses and the function of the different genetic fragments and proteins of this virus that constitute this virus. And as you can see here, we have a single-stranded DNA with two inverted terminal repeats at either end. We have different promoter sequences, rep and cap genes. And in the past 50 years, the AAV virus genetic content and the proteins that are being expressed have been elucidated. And knowing this allowed us actually to pull and tease all these functions apart and to construct 3 plasmids to construct a recombinant adeno-associated vector. These 3 plasmids are, on the one hand, a transfer plasmid, a packaging plasmid, and an adenovirus helper plasmid, all of them encoding the essential information, and only that information required to build a new particle, carrying at this moment, as indicated here, a promoter with a specific transgene of our interest. These 3 plasmids are next transfected into a human producer cell line, for example, HEK, human embryonal kidney cells, but also other cell platforms are available. When we transfect these plasmids, the vector genome will be provided by the transfer plasmid. The structural proteins will be translated from the RNA transcribed from the packaging plasmid, and the helper proteins will be provided by the RNA transcribed from the adenovirus helper plasmid. When in a cell, all 3 plasmids are present at the right ratio, functional adeno-associated viral vector particles will be released either in the cell or in the supernatant. This can be collected, concentrated, and purified, and this will result in a product, a gene therapeutic product that can be applied to our patient to try and reach the cells or the target tissues. Depending on the type of target tissues, we can go with an intravenous application or an intracranial application, for example. These particles will then be provided. The AAV particles will bind specific receptors depending on the serotype of the AAV vectors. There will be endosomal encapsulation, and only those particles that are efficient enough to escape endosomal fusion with lysosomes will reach the nucleus. In the nucleus of the target cell, the second strand will be constructed based on the either the positive or negative single-stranded DNA that is provided in the viral vector. There will be concatenamers formed, and from these double-stranded DNA concatenamers, transcription will take place. There will be export of the RNA, and there will not be any production of additional viral vector particles because that genetic information is completely absent, but instead there will be a transgenic or a recombinant protein produced that is lacking in the cell or tissue of our patient. Similar approaches as described here for adeno-associated viral vectors have also been described for HIV, lentiviruses, and MLV gamma retroviral based vectors platforms. And just to give you in the last few slides an example of a product that is on the market since 2017 in the USA, approved by the FDA, and a year later also in Europe, is the product Luxturna, which is an AAV2 based product for retinal gene therapy to treat a congenital blindness or Leber congenital amaurosis. It's a rare disease, 1 in 50,000 to 100,000 patients, and these patients have or will suffer loss of eyesight over time due to the lack of an RPE65 protein or a functional version of this protein. And the gene therapy consists of the subretinal injection of an AAV2 based viral vector that encompasses the RPE65 cDNA, so the correct version of the cDNA, which upon transcription and translation will provide a functional RPE65 protein. The injection will occur subretinally and thereby transducing the retinal pigment epithelial or the IPE, and this will result in reintroduction of a functional cDNA. cDNA will be transcribed, and this transcribed cDNA will be translated into a functional RPE65 protein, resulting in the restoration of the visual cycle for these patients. And with that, I will now transfer the microphone to Professor Adjali.
Thank you very much, Rick, for this very clear overview. I would like now to introduce our second speaker of today, which is Oumeya Adjali. And while she's loading her presentation, I will shortly give her curriculum. So she graduated as an MT at the University of Constantine in Algeria. And she obtained her PhD, and I know her since then, at the University of Montpellier. She joined the French Institute of Health and Medical Research, INSERM, in 2019, and she's now a senior scientist, a research director, and head of the Translational Gene Therapy Laboratory in Nantes University since 2017, and she has been working at the interface of cell and gene therapies, using as well lentiviral as adeno-associated viral vectors for over 18 years. So she's also a scientific coordinator of the Centre de Production des Vecteurs, so a vector producing unit at Nantes, which now is also an industrial integrator for bioproduction. So please, Mia, go ahead.
So thank you, Els, for the introduction, and thanks to the French Society of Cell Gene Therapy for the invitation. Welcome to all in this webinar. I'm going to follow now after Rick, Professor Gijsbers presentation, who has shown the tremendous potential of gene therapy strategies, to now talk about in vivo based gene therapy using AAV vector and how to make this strategy transferable to the patients and to the clinics. So Rick has already nicely shown the landscape of gene therapy. Oops, so.
It's fine.
And as he has mentioned, there are many vectors. There's no ideal vector, but today AAV, adeno-associated virus vector, represent almost 50% of the strategies under development and evaluation in patients. And when it concerns in vivo-based gene transfer, AAV vectors represent even more than 85% of the strategies. And one reason is that we have at least 30 natural different serotypes for AAV vectors and hundreds of variants. And we can choose, let's say, à la carte, the serotypes based on their tropism, on their cell specificity. And we can also play with different routes of delivery to administer the vector, the therapeutic vector, in the target area, in the target tissue. So as you can see here in the slide, we can inject AAV vectors today via different routes of delivery in the eye, intravitreally or subretinally. We can also target the liver. Yeah. So intramuscular, intravenous, or also in the central nervous system using the intrathecal mode of delivery, or intracerebral, or even in the joint using intra-articular delivery. Now, as Rick has already presented, there are already different AAV products approved in the market. The first one was approved in 2012, but was withdrawn 5 years later. And among the products approved during the last 4 years, 4 of them are injected intravenously using a systemic route of delivery. But to have a therapeutic effect, systemic AAV gene therapy still requires today, with the current vectors, of course, vector engineering is still one important direction, but with the current vector and their therapeutic index, we still need 1 × 10⁻³ to 1 or 2 × 10⁻³ viral genome per kilogram to achieve the therapeutic effect. As you can see, for the 4 products injected intravenously. So as you can see, there's 1 product for Duchenne myopathy, 1 for spinal muscular atrophy, and 2 for hemophilia A and B. And of course, the dose depends also on the disease, and lower doses are needed for hemophilia, for instance, as compared to the muscle or the motoneuron. Now, to make these systemic based gene therapy strategies in the clinics, we need large manufacturing capabilities. And in fact, during years and years, there was a gap, an important gap between AAV preclinical and clinical research and development system and the manufacturing landscape. Because bioprocess development is a very long way to go. First, to produce an AAV vector for an in vivo application, we first need to define the most optimal combination, as already presented very clearly by Rick. First, you have the therapeutic transgene expression cassette, but you also need another plasmid or 2 other plasmids for rep and cap and helper virus functions. You need also to find a permissive cell for AAV production and to define whether you will use a transitory or stable production cell system. And of course, the perfect combination must achieve the highest yield of production with meeting the requirements in terms of quality and safety. The process must be scalable and, if possible, with a reduced cost and a reduced impact on the environment. Now, there are multiple AV manufacturing platforms, but the most used so far, and also for the commercial products, are based on either the use of HEK 293 mammalian cells, either adherent or in a suspension cell culture system, or using the SF9 insect cells, which is a suspension culture system, and using the baculovirus system. So those two systems can be developed for large-scale productions, but to do so, we need to develop a long bioproduction workflow. The production workflow contains different steps. In addition to the plasmid design, plasmid development, plasmid production, there is what we call the upstream process based on cell culture, cell expansion, transfection, and then we can harvest the AV production to purify it, and the purification and concentration step is called the downstream process. Then at the end, we can have a purified and concentrated product that we can formulate and put in the tubes. But as you can see, there are many parameters to assess during the bioproduction process development at each step, and in small scales, and also during the upscaling process. And some of these parameters are not only biological. Some others are biophysical or chemical. For instance, in the bioreactor system culture, we will need also to assess the air rate, also the mechanical forces during the cell culture. Now, to de-risk a process that works perfectly in a small scale, we also need to upscale it and to determine whether we have to change some of the parameters already characterized or set up, but also to de-risk the process and prove that you will have a product with equivalent titers and equivalent quality. And this upscaling in general needs at least a volume of 50 liters, which is already a pre-industrial volume, as you can imagine, which is costly and also time-consuming. Now, the gap I was mentioning before is really narrowed thanks to the different technologies and the different process pipelines that exist today. And because also we have been able to move from either adherent cell-based systems using cell stacks to cell microcarrier-based bioreactors that have greatly expanded the cell attachment surface during the production. But we have also now suspension cell-based systems with large volume bioreactors, and for the AV field, up to 1,000 to 2,000 liters in the industry. Yet, the gap is still there, and we still need really to optimize the AV manufacturing part. And this is a very concrete example to demonstrate this limitation. We're still encountering in the field. So the example is a French product currently under clinical evaluation. It's a Geneton product for Duchenne myopathy in AV8. And the preclinical evaluation was, my laboratory has participated to the preclinical study in the models. So if we consider that, and the studies have shown, and now in the clinical trial, the doses, the escalation dose is between 1 to 6 to 10 to the 13. If we consider that for 5, 10 to the 13 Vg per kilo, and the patient weight is around 30 kgs. If we consider also that we have around 30,000 patients in France, and if we consider that 5, 10 to the 13 Vg per ml is the best yield after purification, even if it's now improving, we will need a large number of adherent cell stacks for adherent cultures, or even hundreds and hundreds of bioreactors of 500 liters, which means that we still need improvements in terms of yield and quality, and also in terms of vector design, to have vectors with improved therapeutic index. And this is why there's, as this webinar is organized by the French Society of Cell and Gene Therapy, I just wanted to mention that in France, there's a French national initiative aiming at accelerating the development of biotherapies and bioproduction pipelines. And one of the objectives is to help academics and industrial players to develop together improved process with higher yields, improved quality, and reduced costs. And among the actors, Els has already mentioned, in Nantes, we are working very hard on the development of new AAV process pipelines at the CPV, Centre de Production de Vecteurs, called now Viral Vector Manufacturing Center, VIVEM, and we will really happy to discuss if you have any question regarding AAV manufacturing. Now, there's not only, we need to produce the product, but first, we need to characterize it before moving to the patient. And I just want to really briefly discuss the issue of quality and AAV analytics, because it is also a very important step during the translation of a product from bench to bedside. So one major limitation today, and even with the current platforms I have shown earlier, is that not all recombinant AAV particles produced today are good products. Yes, it depends on the product, it depends on the process used, but yet we still have full particles with complete AAV genomes, but also some not imperfect particles with no or reduced therapeutic effect. And those particles are either intermediate with a partial encapsulation of the viral genome, or even empty with no viral genome. Another issue is that we do not have only the viral genome we want to encapsulate in the particles, but also some DNA contaminants coming from the vector plasmids, the helper plasmids used, and also from the producer cells. And these DNA contaminants are currently being characterized thanks to high throughput DNA sequencing strategies, which are showing that we can have non-negligible DNA contaminant in the batch, but also sometimes encapsulated in the AAV particle. So we need a full characterization of each AAV batch before moving to the patients, looking at different parameters, safety, identity, purity, potency or functionality, and also, of course, stability a long time. To finish, so we hope that with my colleague Rick, we have demonstrated how much gene therapy is now a real pharmaceutical reality. But I just wanted to finish with one major limitation today, which is the price of those advanced innovative products. Yes, because AV products are the most expensive products so far today. And the last ones approved during the last 2 years cost between 3 million to 3.5 million, which is really very important. And of course, these are innovative products with a very long time for development of the product and also of the manufacturing process. But we really need a sustainable business model also to reduce the cost and make those products accessible to patients, because first of all, gene therapy is a matter of human. And to do so, scientists as well as patients and their families have to play a critical role, and along with industrial and of course politicians. So I will stop here. Thank you for understanding, and we're for listening, and we're ready for the questions.
Thank you, Mia, for this clear presentation. So we can go over to questions. Yes. So I have a question which is for Mia, for example, because I think she's doing a lot of work on that. What we all know and realize is that AV vectors can induce immune responses. So maybe even strong immune responses, and there can already be pre-existing immunity. So how do you deal with that, Mia? How can you avoid that? Can we do a second injection if the first one was not good enough? How is this working?
Yes, your immunogenicity is one other problem with AV vectors, and there are solutions, hopefully. So, of course, high dose vector systemic dosing also increases the risk of this immunogenicity. So it's really something critical to deal with today. So first, no, we cannot administer 2 times these vectors. First, we have a pre-existing immunity, and this pre-existing immunity can neutralize the vectors. So for systemic dosing, for instance, patients today are screened before vector administrations, and only negative patients for antibodies against AAV, so naive patients who have never seen the wild type virus, can be treated. The other thing is that once the vector is injected, administered, we also develop an immune response with neutralizing antibodies, which make a second administration not possible. But there are hopefully solutions. The first one is to reduce this immunogenicity or these immune responses. First, using immunosuppressive regimen drugs, as we have used earlier for allogenic graft rejection, for instance. So we are using, conventional immunosuppressive drugs to modulate this immunogenicity. But there are many other solutions. Some of them are dealing with the vector itself, making it more invisible towards this immune system, playing with the epitopes on the capsid, or inserting some immunomodulatory sequences in the viral genome. And some others are also evaluating some innovative or more, let's say, exotic strategies. For instance, the injection of some enzymes, IDES enzyme, to degrade the pre-existing neutralizing antibodies, or using nanoparticles with immunomodulatory properties. So yeah, there are many developments currently ongoing.
And I have just, because you also talked a little bit about production of AVs, well, Rick as well could answer probably the question, but my question is, if you switch serotypes, because for the brain it's better an AV8 than for another target tissue, it's better to have another serotype. How different is the production process? Do you have to adapt a lot of parameters? do you have to start from scratch to produce these vectors at high titers? How is this working?
No, so I'm sure we'll have a different point of views, or maybe complementary for sure. So no, we do not start from zero, hopefully. So I mean, if we have already an expertise for one serotype of interest, we can adapt the process to another serotype. But the serotypes are different. They have different behaviors during the production. Some of them, for instance, are more secreted or more easily secreted during the production, and some others stay intracellularly in the producer cells. So which means we have to adapt some critical steps, for instance, during the purification or the clarification. Of course, the plasmids are different, as we need the cap gene for the serotype of interest. But yeah, we can adapt the protocols.
Maybe I can add, if that would be fine, Els.
Yes, sure.
Yeah, is indeed, yeah, I follow completely what Mia mentioned. I think from our point of view, we're an academic core where we try to be able to produce on a similar platform a wide variety of serotypes. So in that sense, we produce a bit suboptimally. Everybody will realize that also the transgene you're encoding, or the guide RNA or the microRNA you're encoding, can heavily affect your production efficiency. But yeah, in general, if you know already, and your later stage, you know, this is the serotype I'm going to use, this is the genetic information I'm going to use, there's still quite some room for optimization, doing design of experiments, for example, or even Bayesian models, to really optimize plasmid ratios, transfection efficiency, harvesting, medium replacement regimens, allowing you to push production efficiency of number of particles per cell by 4 to 10-fold, for example. So indeed, when you really would go for the large production efficiencies, these kind of approaches will be very valuable. If you're producing a viral vector for research purposes, most of the time we're not going into in that much of detail in these kind of optimizations.
Then I have for Rick a bit of a general question for so the audience can appreciate the difference really between different vector types, because we use, for example, AAV for liver transduction, and for diseases like hemophilia, retina, we use also AAV, but for hematopoietic stem cell correction, we will not use AAV. Can you elaborate a little bit more why that really is? Because it's, I think, a very important issue, because you said in your at the beginning of your talk, we need to take the right vehicle, delivery vehicle for our therapeutic gene for the right target cell. So
No, I fully agree. So the point basically at the moment is when you're starting and studying a disease, and you want to develop a gene therapeutic approach, one of the first things will be, do you want, or do you require, in order to cure the specific cell or tissue, do you require to go in vivo? For example, if your target is the liver or the brain, that it's not easy to take it out of a patient, treat it ex vivo, and put it back in again. So in those cases, we will mostly use adeno-associated viral vectors. The reason being that we can produce them to quite large quantities, and they spread very easily in whole tissues. If, for example, hematopoietic stem cells or the blood cells are your target, the blood is kind of like a strange tissue. You can consider the whole blood as a tissue, but you can easily take it out. We can manipulate it. I mean, I make it sound all quite easy. I mean, trust me, there are quite some challenges there. But you can manipulate them, select the cells that have now carry your genetic modification, and you can put them back into your patient. And something that's evolving now, actually based on this tissue, the blood that you can take out and put back in, there are several companies, but also academic groups exploring the idea of, for example, taking out a kidney or taking out a liver or part of a liver. It's mostly animal models, to do an ex vivo perfusion and take actually also that opportunity to treat that organ and then put it back in. The advantage there would be, as you mentioned, that the immune response, at least in theory, only the residing immune cells will encounter the viral vector. But that allegedly could lead that patients, for example, suffering from a kidney disease, could be treated either before the phenotype of the disease is occurring, or at least the kidneys possibly could have a longer lifetime into the patients, and helping them in that sense, that kidney transplantation is not that required within a limited amount of time.
And there's one, I think, particular difference is that we use for hematopoietic stem cell better integrative factors.
Yeah, sure.
Because
yeah,
they will proliferate very quickly.
Mm-hmm.
These are not completely, but for the majority, they are not integrating. So
Mm-hmm.
this means that tissues like the eye, where there's almost no division or the liver, that we can use these AAV factors. And of course, there's the difference in size of genes, but that you already mentioned.
Yeah, and then on top of that as well, I mean, in that sense, it's you really have to define in advance what you want to do. If for example, the idea is that you want to, for example, kick out an inhibitor or a repressor, like one of the approaches currently for sickle cell disease, then even a hit and run, for example, providing a quick CRISPR cut, in theory at least. So you could even provide virus-like particles, which we didn't even touch upon here, or you could come in with an RNA therapy to transiently express a protein such as Cas, and to cut a specific repressor to reactivate certain gene programs.
So maybe just one last question to Mia. You talked about the problem of costs and the fact that not all particles, and that's why we're injecting quite huge amounts of vector. I think there's a lot of empty particles that don't carry your gene of interest. I know that people are working on that to try to improve or to reduce the number of empty AAV vectors. Can you just comment a little bit on that? What the strategies might be? I mean, you don't have to mention them all, but just to that people have an idea what is going on. No.
Yeah, so it's indeed a major problem with the AAV production, but not only AAV. So I think the most important direction is to optimize the construction used during AAV production. So people are very much working on the plasmids expressing cap-rep and also the helper function, because this could be improved encapsulation, the correct encapsulation of the viral genome in the particle we are producing. But another thing is that the amount of intermediate or empty particle is really also depending on the technological platform we're using. And now we have enough background. There are also much more potent analytical methods to look at the quality of the particles. And we know that, for instance, SF9 baculovirus-based system leads to much more empty or incorrect, imperfect AAV particles as compared to 293 mammalian cells. So that means that we really need to work on the constructions and the molecular design of the tools we're using to produce AV vectors.
Okay.
And also on the analytical methods to better discriminate full, empty, and intermediate, much more difficult for intermediate particles.
And it seems to be, I mean, something which I find quite amazing. It's quite difficult to move away for different vector systems from 293 T cells, right? To produce better. We tried a lot of things already, but it doesn't seem to be so easy to find another cell type that will release the vectors efficiently and allow pseudotyping, allow all kinds of things. It's quite amazing after all these years that we don't find anything that is really better in a way.
Yeah, that's true.
So I would like to thank you for answering the questions, and again for the really nice talks that you were giving. And I thank also the audience for listening, and I hope to see you for a following webinar. Or I would also advise you to go back to the other webinars on our YouTube channel and on the online site of the SFTCG. Thank you very much, and see you next time.
See you. Cheers.
Thank you. Bye.